So like the unit 9, that is biotechnology, the second last unit. So there are like two chapters. First one is the biotechnology principles and processes. Second one is the biotechnology and its application. So what's the biotechnology principles and process, and in next chapter we are going to study the application. So ever since the days of okay, so this is what's important in here. uh, uh, okay, the whole approach to understanding natural phenomena became anthropocentric. Physics and chemistry gave rise to engineering. Mm, the major utility of the biological world is as a source of food. The major utility of the biological world is as a source of food. Biotechnology, the 20th century offshoot of modern biology, change our daily. No. So, biotechnology, modern biotechnology, improvement in health and food production. Nothing is that important. Okay. So Herbert Boyer, like rDNA technology. Okay. So so what about him? Okay, he performed studies on a couple of restriction enzymes of the E. coli bacterium with specially useful properties. Boyer observed that these enzymes have the capability of cutting DNA strands in a particular fashion, which left what has become known as sticky ends on the strands. These clipped ends made pasting together pieces of DNA a precise exercise. Okay, this is the Herbert. Okay, Herbert Boyer. This is the Herbert Boyer. And what about the Cohen? Cohen had been studying small ringlets of DNA called plasmids. Ringlets of DNA called plasmids, which float about freely in the cytoplasm of certain bacterial cells and replicate independently from the coding strand of DNA. Cohen had developed a method of removing these plasmids from the cell and then inserting them in other cells. Combining this process with that of DNA splicing enabled Boyer and Cohen to recombine segments of DNA in desired configuration and insert the DNA in a bacterial cells, which could then act as a manufacturing plants for specific proteins. This breakthrough was the basis upon which the discipline of biotechnology was founded. So this is Herbert. Remember Herbert, restriction enzymes of the E. coli bacterium, and Cohen, plasmid. About plasmid, he reinserted them in other cells, right? So this is it. Now the biotechnology principles and processes. So biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products, processes useful to humans. In this sense, making curd, bread, wine, which are all micro-mediated processes, could also be thought as a form of biotechnology. However, it is used in a restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on a large scale. Further, many other processes techniques are also included under biotechnology. For example, in vitro fertilization leading to a test tube baby, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene, that is gene therapy, are all part of biotechnology. Now, the European Federation of Biotechnology (EFB) has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural science and organisms, cells, parts, and thereof, and molecular analogues for products and services. So this is the modern technology. Modern biotechnology EFB given this definition, and EFB includes traditional and modern molecular biotechnology, the integration of natural science and organism, cells, parts thereof, and molecular analogues for products and services. Now, principle of biotechnology. Okay, among many, the two core techniques that enable birth of modern biotechnology are genetic engineering. Techniques to alter the chemistry of genetic material, that is DNA and RNA, to introduce into the host organisms and thus change the phenotype of the host organism. So this is the genetic engineering, right? Now bioprocess engineering, maintenance of sterile, that is microbial contamination free ambience in chemical engineering processes to enable growth of only the desired microbe eukaryotic cells in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, and enzymes. It is like contamination free, like it is free of the like pathogens or something like that. It's clean. So this is the maintenance of sterile condition ambience. So So this is the genetic engineering that alters the genetic material, and another process is the bioprocess engineering. So now the conceptual development of the principles of the genetic engineering. Sexual reproduction or asexual reproduction. The former provides opportunity for variation and formulation of unique combination of genetic setup, some of which may be beneficial to organism as well as population. Asexual reproduction preserves the genetic information, while sexual reproduction permits variation. Okay, traditional hybridization procedures used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with the desired genes. But in traditional hybridization there is also useful and unuseful genes also involved. So this technique of genetic engineering, which include creation of, now this is like kind of extra work, like you are also getting useful and undesirable and desirable genes. So the techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation and allows to isolate and introduce only or a set of desirable genes without introducing undesirable genes into the target organism. Okay, so do you know the likely fate of a piece of DNA which is somehow transferred to an alien organism? Most likely this piece of DNA would not be able to multiply itself in the progeny cells of the organism, but when it gets integrated into the genome, okay, the genome, that is nucleus. So if it gets integrated, then there is replication, then this piece is also going to replicate it. So integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. Now this is because the aligned piece of DNA has become part of the chromosome, which has the ability, and chromosome has the ability to replicate. In a chromosome there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. Therefore, for the multiplication of any aligned piece of DNA in an organism, it needs to be a part of chromosomes which has a specific sequence known as origin of replication. Thus an aligned DNA is linked with the origin of replication, so that this aligned piece of DNA can replicate and multiply itself in the host organism. This is also called as the cloning, or making multiple identical copies of any template DNA. Okay, so this is it. Now the construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid that is autonomously replicating circularly extra chromosomal DNA of Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistant genes by cutting out a piece of DNA from a plasmid which was responsible for confirming antibiotic resistance. Okay, accomplished this in 1972 by isolating the antibiotic resistant genes by cutting out a piece of DNA from a plasmid. Isolating the antibiotic resistant genes by cutting out a piece of DNA from a plasmid which was responsible for confirming antibiotic resistance. The cutting of DNA at specific location became possible with the discovery of the so-called restriction enzyme, that is molecular scissors. Okay, the cut piece of DNA was then linked with the plasmid DNA. This plasmid DNA acts as a vectors to transfer the piece of DNA attached to it. Okay, you know the mosquito that acts as an insect vector to transfer the malaria parasite into the human body. In the same way, this plasmid acts as a vector to deliver an aligned piece of DNA into the host organism. The linking of antibiotic resistant gene with the plasmid vector became possible with the enzyme DNA ligase. The linking of antibiotic resistant gene with the plasmid vector became possible with the the linking of antibiotic resistant gene with the plasmid vector became possible with the enzyme DNA ligase, which acts on cut DNA molecules and joins their ends. This makes a new combination of circular autonomously replicated DNA created in vitro and is known as a recombinant DNA. When this DNA is transferred into the Escherichia coli, a bacterium closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistant gene in E. coli was called the cloning of antibiotic resistant gene in E. coli. Okay, now you can hence infer that there are three basic steps in genetically modifying an organism: the identification of DNA with the desirable genes that we really wanted, and then introduction of that identified DNA into the host that can be yeast or bacteria or anything that has a tendency to replicate, maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Maintenance, that is this bioprocess engineering, that is chemical engineering, that maintenance of sterile conditions free from pathogens and all that. Okay, so maintenance of introduced DNA in the host, maintenance of the introduced DNA in the host so that it can replicate, and transfer of the DNA to its progeny. So tools of recombinant DNA technology. First is the restriction enzymes. Then there is the tools of the restriction enzymes are. Then there is like okay, just study like okay. So in here, so the tools of the recombinant DNA, tools of the recombinant DNA technology. Okay, as we know from the foregoing, then that genetic engineering or recombinant DNA technology. So genetic engineering and recombinant DNA technology is the same term. Okay, so they have the key tools like restriction enzymes and polymerase enzymes, and then ligases, then vectors and the host organism. Now first one is the restriction enzymes. In year like 1963, two enzymes responsible for restricting the growth of the bacteriophage in E. coli were isolated. Restricting the growth of bacteriophage, that is the viruses that causes infection to the E. coli in E. coli were isolated. One of these added methyl groups to the DNA while the other cut DNA. The latter one was that cuts the DNA is restriction endonuclease. The first restriction endonuclease is HIN2, whose functioning depend on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. The first restriction endonuclease HIN2, whose functioning depend on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. It was found that HIN2 always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. This specific base sequence is known as the recognition sequence for HIN2. Besides HIN2, today we know more than 900 restriction enzymes that we have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences, right? The convention for naming these enzymes is the first letter of the name comes from the genus, okay, E, that's Escherichia, and then coli. The second two letters come from the species, okay, of the prokaryotic cell from which they were isolated. Now eco RI comes from the Escherichia coli RY30. Now RI, the R is derived from the name of the strain, and Roman number following the name indicate the order in which the enzymes were isolated from that particular strain of the bacteria. So restriction enzymes belong to a large class of enzymes called nucleases. Two kinds, exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make cuts at a specific position within the DNA. So within the DNA is endonuclease, and cuts from the ends, that is exonuclease remove nucleotides from the ends of the DNA, that is exonucleases. Now each restriction endonuclease function by inspecting, like monitoring the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbones. So each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. So each restriction endonucleases so each restriction endonuclease recognizes a specific palindromic nucleotide sequences in the DNA. Okay, now action of restriction enzyme. You can see the enzyme cuts both DNA strands at the same time. At the same time, they cut both the DNA strands, right? You see, this is the vector DNA. Now GAA, TTC, GAA, TCC, they are 5' to 3', 5' to 3, they are palindromic sequence. Now E. coli cut between GNA, GNA on the opposite strand. Okay. Now E. coli cuts the DNA between bases GNA only when the sequence GAA, TTC is present. This is when only it cuts is present in the DNA. So you can see GAA, TTC on both the strands in the antiviral position. So they produce the sticky ends. Now the DNA fragments join as sticky ends. You can see the formation of recombinant DNA. The steps in formation of recombinant DNA by action of restriction endonuclease enzyme, that is EcoRI. Now you see the Malayalam. You can read the same on both the side, in this side and in opposite side, right? You will see the same meaning. Okay, the palindrome in DNA sequence is a base pairs that reads same on the two strands when orientation, but orientation reading is kept the same. We should keep the orientation reading is same, that is 5' to 3' in 5' to 3'. So this is true if read in the 3' to 5' direction. Okay, for example, the following sequence reads the same on the two strands in 5' to 3' direction. This is also true if we read the 3' to 5', okay, on both the sides. Okay, now restriction enzyme cut the strand of DNA a little away from the center. It does not get exactly at the center, but a little away from the center of the palindrome sides, but between the two bases on the opposite strand. This leaves single stranded positions at the ends. So this leaves single stranded positions portions at the ends. So these are overhanging stretches called the sticky ends on each strand. So these are named so because they form hydrogen bonds with their complementary counterparts. This stickiness of the ends facilitates the action of the enzyme, that is DNA ligase. So restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources or genomes. So when cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and this can be joined together end to end using DNA ligases. Okay, so you can see the foreign DNA, same restriction enzyme cutting both foreign DNA and vector DNA at specific point. You see the vector DNA and plasmid, so it cut by the same restriction enzyme. Okay, wait, that is vector DNA and foreign DNA. As you said, ligases join foreign DNA to plasmid. So recombinant DNA molecule is formed. Then there is a transformation. Then cells divides. And that is cloning host, that is E. coli, and then formation of the progeny, and we get the multiple copies of the required desired gene. Also diagrammatic representation of the recombinant DNA technology. Now we are going to study okay, you may have realized that normally unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecules cannot be created. It cannot be created if it is cut by the two different enzymes, two different restriction enzymes, right? Like the vector DNA and source DNA, it should be cut it with the same enzyme, the vector DNA as well as source DNA. So separation and isolation of DNA fragments. So cutting of DNA by restriction endonucleases results in the fragments of DNA. So these fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode. Anode is a positive and DNA is a negative, so it moves towards the anode under an electric field through a medium matrix. Nowadays the most commonly used matrix is agarose gel, which is a natural polymer extracted from seaweeds, right? The DNA fragments separate resolve according to their size through sieving effect provided by the agarose gel. So DNA fragments separate according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, the farther it moves. So smaller the fragment size, the smaller, the farther it moves. The speed it has, the farther it moves. Okay, now we can say guess at which end of the gel the sample was loaded. As you can see, the sample was loaded. A typical agarose gel electrophoresis is showing migration of undigested. Lane 1 has an undigested. Now digested state of DNA fragments, that is 2 to 4. You can see the largest are near the wells and smallest are farther away from the wells. And this is it, like DNA bands and everything. So the separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide followed by exposure to UV radiation. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide followed by exposure to UV radiation. So you cannot see pure DNA fragments in the visible light and without staining. So you cannot see the pure DNA fragments in the visible light and without staining. You need to first stain with ethidium bromide and then further exposed to the UV radiation, then you can see it. So and you can see the bright orange color bands of DNA in a ethidium bromide stained gel exposed to UV light. The separated bands of DNA are cut out from the agarose gel and extracted from the gel. This process is known as the elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with the cloning vectors. Now the second thing is now the cloning vectors. So there are plasmids and bacteriophage. Now the bacteriophages, because of their high number per cell, one cell has more than one bacteriophages, so they have the very high copy numbers of their genome within the bacterial cells. Some plasmids may have only 1 or 2 copies per cell, whereas other may have 15 to 100 copies per cell. So their numbers can go even higher. If we are able to link an align piece of DNA with the bacteriophage or plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. So vectors used at present are engineered in such a way that they help. The vector should be like help easy linking of foreign DNA and selection of recombinants from non-recombinants, right? So this should be the vectors, and it should be like selection of recombinants from non-recombinants and easy linking. So this should be the requirement for the cloning vectors. Now the following are the features that are required to facilitate cloning into a vector. Okay, so origin of replication. This is a sequence from where replication starts, and any piece of DNA when linked to this sequence can be made to replicate within the host cell. So this sequence is also responsible for controlling the copy number of linked DNA. It is equal in number. The copy number will be equal. As the vectors will replicate, the DNA will be replicated. So equal in number. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin support high copy number. If the origin supporting the high copy number, then it will produce the high copy number, right? The vector should be supporting the high copy numbers. Now the selectable marker, in addition to override, the vector requires a selectable marker which helps in identifying and eliminating non-transformers and selecting permitting the growth of the transformer. So selectable marker, in addition to override, origin of replication, the vector requires a selectable marker. The vector requires a selectable marker which helps in identifying and eliminating non-transformers and selectively permitting the growth of the transformer. So transformation is a procedure through which a piece of DNA is introduced in a host bacterium. Okay, normally the genes encoding resistance to antibiotics such as ampicillin. So ampicillin is a gene, then chloramphenicol is a gene, tetracycline is a gene, or kanamycin. No, all these are antibiotics. Two antibiotics such as ampicillin, chloramphenicol, then tetracycline, then kanamycin, etc. are considered useful selectable markers for E. coli. Okay, because the genes encoding resistant to antibiotics such as ampicillin, chloramphenicol, tetracycline, kanamycin are considered useful selectable markers for E. coli. The normal E. coli do not carry resistance against any of these antibiotics. Okay, so the third one is the cloning sites. In order to link the aligned DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzymes. Okay, the presence of more than one recognition sites within the vector will generate several fragments which will complicate the gene cloning. Okay, the ligation of aligned DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. So the ligation of aligned DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. The ligation of aligned DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. For example, you can ligate a foreign DNA at BAM-H1 site of tetracycline resistant gene in the vector PBR 332. As you can see, now we study this diagram. Now first starting with the eco-RI, you can see eco-RI, then PU1, then PST1. See, it has the ampicillin tetracycline resistant gene. Okay, then ORI, then rope has the PUV2. Okay, PU1, PST1 at the ampicillin resistant gene, then ORI, then rope, that is PU2, then SAL1, then BAM-H1 at these are the restriction sites at the tetracycline resistant gene, and then this is the HIND3 not HIND2, and then CLA1. So this is it. Now what is the description is given: E. coli cloning vector PBR 332 showing restriction sites HIND3, eco-RI, BAM-H1, SAL1, PU2, PST1 and CLA1. All these are restriction sites. Okay, now ORI, that means the restriction enzyme will come here and cut it. Okay, now ORI, origin of replication, where the replication initiates, and antibiotic resistant genes, that is ampicillin resistant genes and tetracycline resistant genes, and rope codes for the proteins involved in the replication of the plasmid. Okay, this is it. Now, so when we ligate a foreign DNA at the BAM-H1 site of tetracycline resistant gene in the vector of PBR 332, the recombinant plasmids will lose tetracycline resistance. The recombinant plasmid will lose the tetracycline. The recombinant plasmid will lose tetracycline resistance due to insertion of a foreign DNA, but can still be selected out from the non-recombinant ones by plating the transformers on ampicillin containing medium. So the recombinant plasmids will lose the tetracycline resistance due to insertion of foreign DNA, but can still be selected out from the non-recombinant ones by plating the transformant on ampicillin, because it still has the ampicillin resistance gene, even if the tetracycline resistance gets inactivated. Okay, the transformers growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will grow in ampicillin containing medium but not on the tetracycline because it's inactivated. Okay, because we ligate them at BAM-H1, which has the tetracycline resistant gene, gets inactivated. Now but non-recombinants will grow on the medium containing both the antibiotics. But recombinants, it has the plasmid, so it has the ampicillin resistant gene and tetracycline resistant gene, so it can they can grow on both the medium containing the antibiotics. So In this case, one antibiotic resistant gene helps in selecting the transformer, whereas the other antibiotic resistant gene gets inactivated due to insertion of ln-DNA and helps in selection of the recombinant. So in this case, one antibiotic resistant gene helps in selecting the transformers. Now this is selecting the one antibiotic resistant gene helps in selecting the transformers, whereas the other antibiotic resistant gene gets inactivated due to insertion of ln-DNA and helps selection of the recombinant. So these are the selectable markers. Which are the selectable markers? The gene that encodes resistance to antibiotic, because it helps in selection the recombinant and non-recombinant, and then non-transformant and transformant, all these things. Okay. Now selection of recombination due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating. Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of a chromogenic substrate. In this, a recombinant DNA is inserted within the coding sequence of an enzyme, beta-galactosidase. Now, the gene is in this, a recombinant DNA is inserted within the coding. A recombinant DNA is inserted within the coding sequence of an enzyme, that is beta-galactosidase. Now this results into inactivation of the gene for synthesis of this enzyme which is referred to as an this results into inactivation, which is referred to as an insertional inactivation. The presence of a chromogenic substrate gives blue colored colonies if the plasmid in the bacteria does not have an insert. The presence of a chromogenic substrate gives blue colored colonies if the plasmid in the bacteria does not have an insert. The presence of a chromogenic substrate gives blue colored colonies if the plasmid in the bacteria does not have an insert. Okay. So in the normal case, okay, the presence of insert results into insertional inactivation of the beta-galactosidase gene, and the colonies do not produce any color. So these are identified as recombinant colonies that do not produce any color, that is blue color. Now vectors for cloning. Now vectors for cloning genes in plants and animals. So you may be surprised to know that we have learned the lesson of transferring genes into plants and animals from bacteria and viruses, which have known this for ages. Now how to deliver genes to transfer eukaryotic cells and force them to do what the bacteria or viruses want. For example, Agrobacterium tumefaciens, a pathogen of several dicot plants, is able to deliver a piece of DNA known as tDNA to transfer normal cells into tumor. Okay, so for example, Agrobacterium tumefaciens, a pathogen of several dicot plants, is able to deliver a piece of DNA known as tDNA to transfer normal cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogen. Okay, similarly, retroviruses in animals have the ability to transfer normal cells into cancerous cells. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. Retroviruses in animals have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by the pathogen in their eukaryotic host has generated knowledge to transfer these tools of pathogens into useful vectors for delivering genes of interest to humans. The tumor-inducing plasmid of Agrobacterium tumefaciens has now been modified into a cloning vector which is no more pathogenic to plants but is still able to use the mechanism to deliver genes for interest into a variety of plants. Similarly, retroviruses have been disarmed and are now used to deliver desirable genes into animal cells. So once a gene or a DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial, plant, animal host where it multiplies. So once a gene of DNA fragment has been ligated into a suitable vector, that vector can be TI plasmid, that is tumor inducing TI plasmid of Bacterobacium, has now been modified into cloning vector which is no more pathogenic to plants. Okay, so this is and retrovirus as well in animals, so now it can multiply. So when we put it into the bacteria, plant or animal host. Okay, now the cloning vectors for cloning. Now these vectors we can put it into bacteria, plants or animal host, the host where it multiplies. So vectors for cloning genes in plants and animals. Vectors for cloning genes. The cloning genes mean the genes that we want. These kids are very annoying. Anyways, competent host for transformation with recombinant DNA. So competent host for transformation with recombinant DNA, right? Okay, so the competent host for transformation with the recombinant DNA. Since DNA is a hydrophilic molecule, it cannot pass through the cell membrane. And cell membrane. No, why this is coming, man? So since DNA is a hydrophilic molecule, it cannot pass through the cell membrane. Why? In order to force bacteria to take up the plasmid, the bacterial cell must be first competent to take up the DNA. This is done by treating with a specific concentration of divalent cation such as calcium, which increases the efficiency with which DNA enters the bacterium through pores in its cell wall. The recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on the ice, followed by placing them briefly at 42 degrees Celsius heat shock and then putting them back on the ice. So first that is calcium divalent cation, then ice, then at 42 degrees Celsius heat shock and then ice. So this enables the bacteria to take up the recombinant DNA. This is not the only way to introduce aligned DNA into the host cells. In a method known as microinjection, recombinant DNA is directly injected into the nucleus of an animal cell. Microinjection in animal cell. In another method suitable for plants, cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA in a method known as biolistic or gene gun. Okay, this is the suitable for the plants because they have the cell walls that is rigid. And the last method uses disarm pathogen vectors which when allowed to infect the cells transfer the recombinant DNA. So these are the disarm pathogens, okay, into the host cell. So the process of recombinant DNA technology. Okay, now the isolation of the gene. The first one is the isolation of the genetic material, that is DNA, then cutting of DNA at a specific location, and then there is like insertion of recombinant, then there is like insertion of recombinant DNA into the host cells, then obtaining the foreign gene product. Okay, obtaining the foreign gene product, and then the last process is the downstream processing. Okay, so these are the processes involved in the biotechnological principles and processes. So the process of recombinant DNA technology. Recombinant DNA technology involves several steps in specific sequence such as isolation of DNA, fragmentation of DNA by restriction endonuclease, then isolation of a desired DNA fragment, then ligation of DNA fragment into a vector, transferring the recombinant DNA into the host, then culturing the host cells in a medium at large scale, and then the extraction of the desired product. So isolation of the genetic material, like okay, DNA along with other macromolecules such as RNA, proteins, polysaccharides and also lipids. This can be achieved by treating the bacterial cells, plant or animal tissue with enzymes such as lysozymes. So in bacteria they break the wall, then cellulase in plant cells, and chitinase in fungus, that is chitin. You know that genes are located on a long molecules of DNA intertwined with proteins such as histones. The RNA can be removed by treatment with the ribonuclease, whereas proteins can be removed by treatment with protease. So proteins can be removed by appropriate treatment with protease. Other molecules can be removed by appropriate treatments and purified. DNA ultimately, so other molecules can be removed by appropriate treatments and purified DNA ultimately precipitates out after the addition of the chilled ethanol. This can be seen as collection of fine threads. As you can see, this is the spooling. DNA that separates out can be removed by spooling. As you can see, this is chilled ethanol is added. Now second step is the cutting of a DNA at specific location. So restriction enzyme digestion are performed by incubating purified DNA molecules with the restriction enzyme at the optimal conditions for that specific enzyme. Agrogel electrophoresis is employed to check the progression of a restriction enzyme digestion. DNA is a negatively charged molecule, hence it moves towards the positive electrode, that is anode. The process is repeated with the vector DNA also. The joining of DNA involves several processes after cutting the source DNA as well as the vector DNA with the same specific restriction enzyme. The cutout gene of interest from the source of DNA and the cut vector with the space and mix and ligase is added. This results in preparation of recombinant DNA. So formation of the recombinant DNA in here. Now the next step, that is amplification of gene of interest using PCR. So PCR stands for polymerase chain reaction. In this reaction, multiple copies of the gene or DNA of interest is synthesized in vitro, that is outside, using two sets of primers, that is small chemically synthesized oligonucleotides that are complementary to the regions of DNA, and the enzyme DNA polymerase. Okay, the enzyme exchanges the primers using the nucleotides provided in the enzyme exchanges the primers using the nucleotides. The enzyme exchanges the primers using the nucleotides provided in the reaction, and the genomic DNA and the genomic DNA as template. If the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times. That is 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase that is isolated from the bacterium Thermus aquaticus, which remain active during the high temperature induced denaturation of double stranded DNA. High temperature that induces the denaturation of the double stranded DNA. The amplified fragment, if desired, can now be used to ligate the vector for further cloning. Now, fourth step is the insertion of the recombinant DNA into the host cell or organism. So several methods of introducing the ligated DNA into the recipient cells. Recipient cells, after making them competent to receive, take up DNA in the surroundings. So if a recombinant DNA bearing gene for resistance to an antibiotic, example ampicillin, is transferred into E. coli cells, the host cells become transformed into ampicillin resistant cells. Now the E. coli becomes transformed into ampicillin resistant cells. If we spread the transformed cells on a agar plate containing the ampicillin, only transformed cell will go understood. Recipient cell will die. So the untransformed recipient cell will die because they don't have the antibiotic resistant gene. Since due to ampicillin resistant gene, one is able to select. Now this ampicillin resistant gene or tetracycline resistant gene, these are able to select a transformed cell in the presence of the ampicillin or tetracycline, whatever. The ampicillin resistant gene in this case is called as a selectable marker. So selectable marker, these are the ampicillin resistant gene or tetracycline resistant gene. Now obtaining the gene product. Okay, so what is the recombinant protein? If any protein encoding gene is expressed in a heterologous host, I mean, if you are trying to E. coli protein into the bacteria, then so it is called the recombinant protein because it is expressed in a heterologous host, that is bacteria. It was protein of the E. coli. Okay, so how this is line tells, if any protein encoding gene is expressed in a heterologous host, it is called the recombinant gene. The cells harboring cloned genes of interest may be grown on a small scale in a laboratory. Now the cultures may be used for extracting the desired protein and then purifying it by using different separation techniques. Now the cells can also be multiplied in a continuous culture system wherein the used medium is drained out from one side while the fresh medium is added from the other side to maintain the cells in their physiological most active log or exponential phase. This type of culturing method produces a larger biomass leading to a higher yields of desired proteins. Now small volume cultures cannot be yield appreciable quantities of products. To produce in large quantities, the development of bioreactors where the large volumes, that is 100 to 1,000 liters of culture can be proceed was required. Thus the bioreactors can be thought of as vessels in which raw materials are biologically converted into specific products, individual enzymes, etc. Okay, using microbial, plant, animal or human cells. Okay, a bioreactor provides the optimal conditions for achieving the desired product. Okay, provides the optimal condition for achieving the desired product by providing optimum growth conditions. Examples, that is temperature, pH, substrate, salts, vitamins and oxygen. Okay, now the simple steered tank bioreactor and a sparse steered tank bioreactor through which the sterile air bubbles are sparged. You see, first case, that is water, foam, beaker, flat bladed impeller, then there is a culture or broth, then there is a sterile air, then acid waves for pH control, then steam for sterilization. In the second term, that is sparse steered tank bioreactor through which the sterile air bubbles are sparged. Okay, so increase surface area for oxygen transfer. Okay, gas entertainment. Okay, bubbles dramatically increases the oxygen transfer surface. So these bubbles increases the oxygen transfer surface. You can see it increases the surface area for oxygen transfer. Now steered. So now a steer tank bioreactor is usually cylindrical or with a curved base to facilitate the mixing of the reactor content. Why it is cylindrical or curved at the base? To mix the reactor contents. Okay, the steerer. Now the steerer facilitates even mixing and oxygen availability throughout the bioreactor. Now the steerer, that is it's kind of a spoon. Steerer facilitates even mixing and oxygen availability throughout the bioreactor. Okay, alternatively, air can be bubbled through the reactor. If you look at the figure closely, you will see the bioreactor has an agitator system. It has an agitator system like agitator system, an oxygen delivery system, and a foam control system, and a temperature control system, pH control system, sampling ports. And sampling ports are also present so that the small volumes of the culture can be withdrawn periodically. So this is a steer tank reactor. Okay, just the only difference is that they have the sterile bubbles in the second diagram. That is past steer tank reactor. Simple steer tank reactor have agitator system and all that. Okay, now the downstream processing. Okay, so that after the completion of the biosynthetic stage, the product has to be subjected through a series of processes before it is ready for marketing as a finished product. Okay, so okay so the downstream. That is after completion of the biosynthetic stage. After completion of the biosynthetic phase, that is all the stage before the downstream processing, the product has to be subjected. The product has to be subjected through a series of processes before it is ready for marketing as a finished product. Okay, the processes include separation and purification, and this downstream processing includes the separation and purification, which are collectively referred to as the downstream processing. The product has to be formulated with suitable preservatives. Now the product has to be formulated with suitable preservatives. Each formulation has to undergo through clinical trials, as in case of drugs. So there are clinical trials for in case of drugs. Now strict quality control testing for each product is also required. The downstream processing and quality control testing vary from product to product, and downstream processing, it varies from product to product. Okay, quality and all that. So this is all, your chapter is done within an hour, including the summary exercises and everything, from introduction and to everything. So obtaining the foreign gene product and all the insertion of recombinant DNA into the host cell and organisms. So this is left, like, thank god I just scroll up and I found this, that I didn't explain this. So the amplification of gene of interest using PCR. Okay, PCR stands for polymerase chain reaction. In this reaction, multiple copies of a gene or DNA of interest is synthesized in vitro using two sets of primers. Okay, that is small chemically synthesized oligonucleotides that are complementary to the regions of DNA and the enzyme DNA polymerase. Okay, the thermostable DNA, that is thermus aquaticus. Okay, high temperature. Now first step is the denaturation, then the annealing, that is the primer annealing. Okay, that extends the nucleotide and makes the copies. Then there is a extension. So you can see first step is the denaturation, then the annealing, then there is a extension, and amplified 1 billion times. You see, heat first. In denaturation there is heat, then annealing, adding of the primers. You see, 3 dash to 5 dash, 5 dash to 3 dash, primers are added. Okay, then DNA polymerase, that is tag polymerase. Okay, thermus aquaticus, right, most thermostable, plus deoxyribonucleotides, that is nucleotides are added in. So DNA polymerase, tag polymerase, and deoxyribonucleotides. Then there is extension. So denaturation, you see double standard DNA gets separated. So denaturation, annealing, and extension. Now polymerase chain reaction, that is PCR. So each cycle has 3 steps, that is denaturation. So polymerase chain reaction, that is PCR, each cycle has 3 steps. That is, first one is the denaturation, second is the primer annealing, that is adding of the annealing, and then the extension of the primers, and third step is the primer extension. So extension of the primers. Okay, so this is it. So cutting of DNA at specific locations, right? So this is all competent host for transformation with recombinant DNA. You see, these 4 points, that the following are the features that are required to facilitate cloning into a vector. What is required to facilitate cloning into a vector? Vector should have the origin of replication, then it should have the selectable marker, then it should have the cloning sites. Okay, and then the vectors for cloning genes in the animals and plants and animals, right? थोड़ा ये भाई उस बनने ने पूरे लोग उसी के बाद जाते थे, पढ़ाता ही था वो वैसे खत्म नहीं. So the principles of the principles of biotechnology, that is genetic engineering and bioprocess engineering. Okay, so it is all like the context where like principles of biotechnology, then tools of recombinant DNA technology, then process of recombinant DNA technology. So all this. Why this kid is crying? Parents are really unaware of these kids, right? They don't care. Why do they give the birth? I don't know. Anyways, your chapter is done. That's it.